Assalamualaikum to all my beloved students. Now we are here continuing our chapter 5 genetics part 4. Gene disorder diseases can be identified at an early stage through amniocentesis and karyotyping. Amniocentesis is a diagnostic technique to identify fetal cell abnormalities or infections as well as for sex determination. It is usually done from the 50th to the 60th week of pregnancy. The purpose of amniocentesis is to diagnose gene disorder diseases such as Down syndrome and sickle cell anemia. This is how the procedure of amniocentesis takes place. At the beginning, the ultrasound technique is used to determine the position of the fetus in the uterus. A needle is inserted very carefully through the mother's abdomen, penetrating the uterus wall. About 20 ml of amniotic fluid is extracted. Then it is centrifuged to separate the amniotic fluid and the fetal cells. Finally, the separated fetal cells will be used to get the karyotype. The risks of amniocentesis are amniotic fluid leakage, injury to the baby, or miscarriage. Meanwhile, the results can be gained from 3 days to up to 4 weeks. Gene disorder diseases can also be identified through karyotyping. Karyotyping is the process by which photographs of chromosomes are taken to form a karyotype. Karyotyping is done to determine the number of chromosomes and detect any abnormalities. Many people use karyotyping to ensure that their unborn babies are healthy. The purpose of carrying out karyotyping is to identify and evaluate the size, shape, and number of chromosomes in a sample of body cells. To do a karyotype, a doctor will need a sample of cells from the patient. Once the doctor has a sample of cells, they need to be grown in the lab in a culture to increase the number of cells available of the cell through mitosis. When cells are in metaphase stage, the chromosomes are condensed. The cells are arrested in metaphase using a chemical called colscamate. The cells are then concentrated and a fixative is applied for preservation. Next, the cells are stained with a dye which helps scientists to see the chromosomes clearly. Lastly, the cells are spread on a slide that can be viewed under a microscope. The pictures are cut and arranged into a karyotype showing all the chromosomes lined up in order. The risks of karyotyping are bleeding, infection, and miscarriage. Meanwhile, the results can be obtained from 1 to 2 weeks. A gene therapy is an example of genetic research which has been carried out for the benefit of humans. Gene therapy is designed to introduce genetic material into cells to make a beneficial protein. The flow of process for gene therapy is divided into three stages: stage A, stage B, and stage C. Firstly, in stage A, the cells are removed from the patient. Next, in stage B, a virus is altered to stop its growth, and a gene is inserted into the virus. In the third stage, stage C, the altered virus is mixed with the cells. The cells become genetically altered. The altered cells are then injected into the patient, and the desired protein or hormone is produced. If a mutated gene causes a necessary protein to be faulty or missing, gene therapy may be able to introduce a normal copy of the gene to restore the function of the protein. Have you ever heard about genetic engineering? Yes, genetic engineering is the term used for genetic modification of an organism. Recombinant DNA technology is one of its examples. Recombinant DNA technology is a technology that combines two different species to produce a new genetic characteristic. Let's look at the making of insulin through recombinant DNA technology. Number one, human insulin is extracted from pancreas cells and an insulin producing gene is isolated. Second, a plasmid DNA is extracted from a bacterium and cut with restriction enzyme, forming plasmid vector. Thirdly, human insulin producing gene is inserted into the bacterial plasmid vector to form the recombinant DNA of human insulin producing gene. Number 4. This recombinant DNA is introduced into a bacterial cell to form the recombinant bacterium. Number 5. The recombinant bacterium multiply in a fermentation tank producing human insulin. And finally, number 6, insulin is extracted, purified, and bottled. It is now ready to be injected into diabetic patients. Genetic engineering is also related to genetically modified organism, GMO. GMO is an organism. It can be plant, animal, bacteria, or virus, which has been genetically modified for a particular reason and purpose. GMO can be done by combining an animal gene with a plant gene or a bacterial gene. This will produce a genetically modified organism with new characteristics that might not be possible to be produced through the traditional crossover process. This application can be seen in paddy, maize, and palm oil cultivation. Bigger fruits, higher nutritional value, and resistance to pests and diseases are the expected characteristics of the product desired in the agricultural field. Genetic engineering technology comes with some advantages and disadvantages. Therefore, it is important for scientists to apply the correct ethics and scientific values in genetic engineering technology with high integrity to avoid them from being not morally and religiously wrong. Thank you.